You run the sample, wait for the sequence to finish, and look up to see the monitor flashing a harsh red 82% fail, when the strict requirement is a tight 95 to 105% and 82 is an immediate red alert. In pharmaceutical manufacturing, this is known as an out-of-specification result, or OOS. The moment it happens, the clock starts ticking. The FDA demands a prompt, thoroughly documented resolution to ensure patient safety. Before anyone panics, analysts initiate Phase 1A. This is the rapid response checklist designed to catch immediate, visible mistakes in the laboratory. We start with errors that are often easy to spot. Did someone shift a decimal point in the math? Is there a visible puddle forming under a leaking HPLC column? Did the solution prep turn cloudy when the standard operating procedure says it should be crystal clear? If those Phase 1A checks come there clean, and our 82% assay is a mystery, we cannot assume the batch is bad. Instead, we must move into a deeper investigation to determine the true source of the deviation. Phase 1B transitions the investigation into a rigorous forensic audit of the entire testing environment to see if a hidden error skewed the data. We use the 6M's Fishbone diagram, where the central spine leads to the failed 82% result. We examine man, machine, and methods for training, calibration, and procedure clarity. Then we check materials, Mother Nature, and measurement for expired reagents, humidity, and standard accuracy. There is a hard rule during this audit. You can never simply point a finger at analyst error to close the book. The FDA requires concrete, documented proof before blaming the human operator. When a meticulous 6M's audit clears the laboratory of any fault, we reach a critical pivot point. If the test itself was flawless, the failure likely originated before the sample even reached the bench. The investigation now leaves the laboratory and enters Phase 2. We expand our perimeter onto the heavy machinery of the manufacturing floor. We search for hiccups in the batch's production, auditing the mixing records, scanning for unexpected temperature spikes, and checking for personnel shift changes. Our logic tree diverges here. The green path is hypothesis testing, a controlled experiment. The red, crossed-out path is illegal testing into compliance. In hypothesis testing, retesting uses the original small vial, while resampling takes a completely new cut from the large manufacturing vat. Distinguishing between testing to discover a root cause and testing to hide an inconvenient result is exactly how a company survives a stringent FDA audit. Let's apply that Phase 2 hypothesis testing to our mysterious 82% floating assay. We suspected an issue with the powder blending machinery on the factory floor and designed a controlled experiment to check it. This animated 3D cutaway reveals the breakthrough. The powder mixes dynamically throughout the main chamber, but notice that stagnant clump trapped in the corner. That is a physical dead spot in the industrial machinery. Because that corner of powder never fully integrated, the overall batch was non-homogeneous. The active ingredient wasn't evenly distributed, which explains why the final tablet we tested only contained 82% of what it should have. No laboratory didn't make a mistake. The test functioned exactly as intended, acting as an early warning system that caught a critical manufacturing defect. With the root cause proven, the consequences are immediate. The out-of-specification result is declared entirely valid, and the defective batch is rejected. From there, we initiate a corrective and preventive action, or CAPA. Manufacturing cannot resume until that blender is physically modified to eliminate the dead spot. By following this path, the investigation avoided the major regulatory pitfalls FDA inspectors actively search for. Looking at this stylized FDA warning letter, you can see the actions that commonly trigger citations. You cannot average a failing score with a passing score to fake compliance, and you cannot let an investigation sit open for more than 60 days without justification. Instead, we followed the continuous flow mapped out on this chart. Identify the issue, execute a Phase 1 lab audit, escalate to a Phase 2 manufacturing review, pinpoint the root cause, and finally implement the CAPA. A rigorous out-of-specification investigation process is a vital defense mechanism. It ensures patient safety and maintains the integrity of the entire